So I'm pleased to introduce today's speaker, Emily Scott. Emily is from Virginia and graduated from the University of Virginia in 2024 with a BS in biology. During her undergraduate years, she traipsed across botanical gardens and field sites while working on the evolution of floral and reproductive traits of species in the bellflower family. And she was also a land management intern with the Smithsonian National Zoo in Conservation Biology Institute. Emily arrived at Archbold in the days after Hurricane Milton and was particularly interested in gaining experience in rare plant conservation. Her independent project, which you're going to hear about today, considers the potential threat of invasive species on rare plants. And specifically, she'll be sharing her work on how road modification affects the competitive ability of natal grass, which is one of the most widespread invasive plants found here in the Florida scrub. Her study is a nice integration of competition experiments, environmental gradients, and field validation that sheds light on how natal grass invades the effects it has on rare plants in the scrub. So after her Archbold internship is over next week, she'll be heading to Longwood Garden in Pennsylvania for one more internship before applying to graduate school. And with that, I'll hand it over to Emily.
Thank you for that introduction. Yes, hi, my name is Emily Scott, and I'm here to tell you all about my intern project entitled Using Ecological Theory to Understand How Road Modification Facilitates Invasive Natal Grass, also known as Melinis repens. Natal grass was first brought to Florida in the late 1800s as an ornamental and forage plant. Today, it is often found in disturbed areas such as along roadsides, in citrus groves, and even here at Archbold, outside of some of our main buildings, including this one. It is designated as a Category 1 invasive plant by the Florida Invasive Species Council, meaning it is capable of causing ecological damage, and studies have found that upon its invasion, there is a decline in native species diversity. Invasive species such as natal grass can often be found along roadsides in the scrub. Roadsides can often have heightened soil pH as a result of human activity like disturbance and road modifications like added shell. Studies have found that this heightened soil pH may explain the occurrence of invasive species found there. In an environment like the scrub, soil pH may be a factor in dictating where invasive species are and are not found. Interior undisturbed scrub has very acidic soil. It can be as low as 4.2. Having the heightened soil pH along the roadsides may explain why invasive species can be found there, but not invade the scrub. However, native species can also be found along our scrub roadsides. Many native species require open gaps in the scrub, and roadsides are able to mimic these open spaces. Here we have a roadside in the scrub with Eryngium cuneiform. Eryngium cuneiform is a rare Highlands County endemic. In this picture, as you can see, it is being surrounded by natal grass. As natal grass invades a roadside, it can come in contact with native species such as Eryngium and cause interspecific competition. Interspecific competition is the competition for space and resources between individuals of different species. As the invasion continues and natal grass becomes more dense, intraspecific competition can occur, which is the competition for space and resources by individuals of the same species. These competition dynamics can dictate if species are able to coexist with each other. Coexistence is thought to occur when intraspecific competition limits a species more than interspecific competition does. However, changes in environmental factors such as resource availability or soil pH can change the existing competition dynamics. As we continue to alter roadsides with things like added shell and increase the soil pH, we may be disrupting the existing competition dynamics and cause our native species to become limited by interspecific competition with natal grass. To understand the threat that road modifications and natal grass play on native species in the scrub, I asked, do road modifications facilitate natal grass performance? Because soil pH is expected to increase with road modifications, I specifically asked, does soil pH influence natal grass germination and biomass? And does soil pH alter competition between the tallgrass and native species? For my third question, I wanted to know if the results that I found in my first two questions translates to the patterns that we see in the field. And so I asked, do road modifications influence soil pH and in turn, the tallgrass growth in the field? Here are my hypotheses. For my first question on if soil pH will enhance the tallgrass performance, I expect that as soil pH increases, so does the tallgrass performance. And then for my second question on if soil pH will alter competition between the tallgrass and native species, I expect that at a low pH, the tallgrass will be limited by interspecific competition, so competition with the native species, while at a high pH, the native species will be limited by interspecific competition, so the competition within the tallgrass. And so now let's jump into my first question of, does soil pH increase the tallgrass germination and biomass? To answer this question, I germinated and grew in the tallgrass seeds that I had collected from the field in different soil pHs. For each cell in my tray, I had 3 tallgrass seeds, and for each pH treatment, I had 10 replicates. I manipulated the soil pH by adding fixed amounts of lime to soil that I had collected from undisturbed scrub. Here are the amounts that I added to my soil of lime, and here are where my pHs ended up. At the bottom, we have my no lime added soil that had a pH of about 4.5, and then at the top, when I added 3 grams of lime, my soil pH ended up at about 7. Once I began the experiment, I then conducted daily germination and survival checks. And then about a month and a half later, I harvested, dried, and weighed all of my seedling biomass. And so now let's go start with my results. I found that soil pH does significantly increase the tallgrass germination. As you can see, as soil pH increases, so does the tallgrass germination. I also found that soil pH significantly increases the tallgrass biomass. Once again, as soil pH increases, so does the tallgrass biomass. And so with these results, I can say that yes, it does appear that soil pH increases the tallgrass performance because as soil pH increases, so does the tallgrass germination and biomass. And so now looking at, does soil pH alter competition between the tallgrass and native species? To answer this question, I grew the tallgrass with 4 different native scrub species, Baldwina angustifolia, Orangium cuneiform, Lechia decurtii, and Lechia cernua. I then had 2 soil pH treatments, one where I didn't add lime and then one where I did. And then for my interspecific competition treatment, I grew natal grass with one of my native species. So it was natal grass and Orangium, or natal grass and Baldwina, and so on. And for each interspecific pair, I grew it in both my no lime and my lime treatment. I then had an intraspecific competition treatment where I grew natal grass with itself, as well as each of my native species with itself. And once again, for each intraspecific pair, I grew it in my no lime and my lime treatment. For each of my pH and competition pairs, I had 10 replicates. So across all of my species and all of my replicates, I had a total of 180 pots going. And here is the same pH to added lime regression as my first experiment, but I added the pHs of the treatments that I used in this experiment. At the bottom, I used the same no lime added treatment as I did in my first experiment. And so once again, the pH was about 4.5. And then my added lime treatment ended up having a pH of about 6.3. And then I had the same procedure and timeline as I did for my first experiment. So starting off with daily germination survival checks, and then about a month and a half later, I harvested, dried, and weighed all of my seedling biomass. I then used my biomass data to calculate the natural log of relative yield. Here, Y just means yield, which is the average biomass of all individuals of a species in a particular treatment. So the equation in this case is the natural log of the average biomass of individuals in a mixture divided by the average biomass of individuals in a monoculture. My individuals in a mixture are experiencing interspecific competition, while my individuals in the monoculture are experiencing the intraspecific competition. If the natural log of relative yield is less than 0, then that means the average biomass of individuals in a monoculture is greater than the average biomass of individuals in a mixture, and biomass is being limited by interspecific competition, or there's some kind of benefit being gained by being grown with itself. If the natural log of relative yield is equal to 0, that means these two values are about the same, and there is no difference in competition. If the natural log of relative yield is greater than 0, then that means the average biomass of individuals in a mixture is greater than the average biomass of individuals in a monoculture, and biomass is being limited by intra-specific competition, or it is gaining some kind of benefit by being grown with other species. And so now starting with my results for this question, this is the germination proportion of all of my species, not separating between pH or competition treatments. As you can see, a lot of my native species did not germinate super well. Because of this, moving forward in my analysis, I decided to lump all of my native species together, and so I will simply be comparing the tall grass to all of my native species as one. And so now starting off with tall grass germination, I once again found that soil pH significantly increased the tall grass germination. This can be seen within each of my competition treatments. As you go from the low pH treatment to the high pH treatment, you see an increase in the tall grass germination. However, I did not find that competition type had a significant effect on the tall grass germination, and you can see that looking between the competition treatments within each pH, and there's similar values. And then for my native species, I found that neither soil pH nor competition type had a significant effect on native species germination, and this can be seen in my plot in the similar values across all of my treatments. And now for biomass, when I did my biomass analysis, I made sure only to include interspecific pairs where at least one individual from each species had germinated, and intraspecific pairs where at least two individuals had germinated to make sure there is an effect of competition occurring. And so for the tall grass biomass, I found once again that soil pH significantly increased the tall grass biomass. Again, looking within each competition treatment, going from the low pH to the high pH, you see an increase in the tall grass biomass, but competition type did not have a significant effect. As you can see, if you look within each soil pH, there are pretty similar values between the competition treatments. And then for my native species, again, soil pH did not have a significant effect on native species biomass, but competition type was found to be marginally significant. If you look within the low pH between inter and intraspecific competition, there are pretty similar values, but when you go to the high pH, you see a little bit of a bump going from inter to intraspecific competition. And now for my relative yield results, here is the equation as a reminder. It is the natural log of the average biomass of individuals in a mixture divided by the average biomass of individuals in a monoculture. If the value is above 0, then that means the average biomass of individuals in the mixture was greater, and biomass is being limited by intraspecific competition. And if the value is below 0, then that means the average biomass of individuals in a monoculture is greater, and biomass is being limited by interspecific competition. So now starting off with the tall grass at a low pH, there is a positive value indicating that the tall grass is being limited by intraspecific competition. This is not what I had initially expected. At a low pH, I had expected that the tall grass would be limited by the competition with the native species, but that's not what I found. For moving on to natal grass at a high pH, there is a little bit of a negative value, but it's pretty close to 0. And so overall competition the difference in competition types does not seem to have an effect on natal grass at the high pH. And now for native species at the low pH, there is a negative value. And so this indicates that at a low pH, native species are limited by interspecific competition. So the competition for the tall grass. But we move on to the high pH, we see a greater negative value, indicating that as pH increases, this effect that the tall grass plays on native species seems to increase. And so using these results, coming back to my hypothesis, we did not find that natal grass was limited by interspecific competition. Instead, it was limited by intraspecific competition. But we did see that at a high pH, native species were limited by interspecific competition, even more so than they were at the low pH. This indicates that natal grass may not be limited by other species regardless of the pH, However, native species are limited by natal grass at the high pH, indicating natal grass still likely has some sort of competitive advantage as the pH increases. This also indicates that because we are seeing interspecific competition at the high pH, coexistence between the species is less likely to occur as we increase the soil pH. And so as a reminder for my last question, I wanted to see if the results of my first two questions translates to what we see in the field. And so I asked, do road modifications influence soil pH and in turn, natal grass growth in the field? And so to do this study, I went to 3 properties, Archbold Biological Station, Carter Creek, and then Sun and Lake / Silver Lake. Those are technically 2 different properties, but I surveyed Sun and Lake and Silver Lake as one. And so for the sake of this study, they are one property. At each property, I surveyed 2 sand and 2 shell roads. In my study, the shell roads are what I am considering modified roads because the shell is added to them. At each road, I laid out a 30-meter transect, and then along that transect, I had 4 2x1 meter quads randomly distributed. And then at each quad, I collected all above ground natal grass biomass as well as a soil sample for pH. And so here are my results. I found that soil pH did have a significant, that road treatment did have a significant effect on soil pH. As you can see, as we move from sand roads to shell roads, we see an increase in soil pH. However, I did not find that road treatment had a significant effect on natal grass biomass. As you can see across the different properties, there's a little bit more of an inconsistent pattern. Here at Archbold, I even found that there was more natal grass on sand roads. That's partially because one of the sand roads that I surveyed at was pretty disturbed, and it was adjacent to a shell road that had an ongoing natal grass invasion that had spread over. And so that just shows sand roads can also be vulnerable to dense natal grass invasions. And then I also wanted to look at the relationship between natal grass biomass and soil pH, and I found a marginally significant one. As you can see, as soil pH increases, so does natal grass biomass slightly. And so overall for my study, I found that soil pH does facilitate natal grass performance, as because when soil pH increases, so does natal grass germination and biomass. I also found that soil pH does appear to alter competition, as when soil pH increases, native species become more limited by natal grass presence. And then based on my field study, I found limited evidence that soil pH dictates natal grass biomass, and therefore soil pH may have less of a role in determining natal grass growth later in its life. And so from my study, it is clear that increased soil pH from road modifications may hurt native species while helping natal grass, and therefore facilitate natal grass invasion into habitats. And so road modifications are not recommended to maintain native species diversity. I would like to thank everyone who supported me, gave me information, and helped me through my project, and I would be happy to take any questions. Yeah.
So how does pH affect plants? Like is it just, is natal grass just calcium limited? Like why does natal grass grow better when you add calcium carbonate?
I think it has to do with the ions and the available ions is kind of my general understanding. But yeah, it overall is, yeah, sorry.
No, I mean, no, that's a good answer. Because if you don't find the same effect in these other three other spots that you saw, then is the high pH there because of some other element that's driving up the pH, but not something that is a nutrient that's limiting natal grass growth? Or
yeah, I mean, that's definitely a possibility. There's a million and one things that people have studied in relation to tall grass and or not just invasive species in general, and what promotes invasive species and habitats. And I focused on soil pH, but it's very likely that there's other things going on that I wasn't able to totally account for in my study, but could also be driving the tall grass, especially on sand roads and stuff.
Yeah, although I think that the relationship between where at high pH, the intraspecific goes away, and at high pH, natal grass is a much better competitor with native species are really cool.
Yes.
I thought it was really cool to see the lower soil pH in the shell rock treatments between Carter Creek, Archbold, and Silver Lake Sun and Lakes. Knowing the history that I do of the area, like the shell rock was placed on Carter Creek like a long time ago, and some of the other locations had much more recent additions, and so seeing, I'm just extrapolating the decline or like the return back to low pH. And just makes me think, is there anything that we can do as land managers when we have to apply Shell Rock Road that might help suppress natal germination and promote the other species outside? You know, should we focus on like an inert fill or maybe planting something or just hose it down with acid?
Yeah, I feel like it's probably just a manual removal because natal grass, it definitely like builds that seed bank, and then like once you like disturb the area and you like bring them back to the surface, then they're able to germinate. So I think just probably manual control is going to be your best bet.
Other questions?
There's one online from Greg Saunier. It appears your natal biomass were pretty low. Do you think the inter versus intra-specific patterns would have been different if you had waited longer to sacrifice your plants?
I mean, I'm sure. Like I only have a limited amount of time here, and there was a little bit of trial and error before I was really able to get started with my project, and so that also limited how much time I was able to have. Like I would have loved to have been able to continue it and seeing if my patterns continue or if maybe things change. Because I did see kind of different results in the field once in a tall grass is like fully developed, it definitely kind of makes me think like things might change from like the early life stages as it like gets bigger and more established like in its habitat.
One.
I was wondering, when you created this project, so like pH, like you kind of mentioned, is related to nutrient availability and other things, and it seems like a lot of these scrub species, the native ones, don't really germinate by seed. To my limited understanding, they grow vegetatively or whatnot, and the tall really seems to grow well from seed. I don't know what question is. I guess, why did you focus on pH in particular? Like, where did that question?
It was mostly due to why. So I started this project thinking about like, okay, we see natal grass like on the roads, but not really in the scrub. Like, why is that? And based on my reading, like soil pH kept coming up in terms of like, well, soil pH along roads, like increases. And so I wanted to see, okay, like, is the soil pH helping? But yeah, like you said, there's all these other like factors of like available nutrients and stuff that could also be influencing where the natal grass isn't found. Yeah, does that answer your question? Sorry. Yes.
Great. I was just wondering how you picked your native species. Are they like also commonly found on roadsides? Do you think that they're like good competitors with natal grass, or how did you pick them?
Yeah, I should have mentioned that in my presentation. But yeah, before I began my experiment, I did a bunch of surveys just kind of on the roads around Archbold to see what native species are found along roads and also what native species seem to be found in areas that could have natal grass or has natal grass. And so that's how I ended up with my 4, is they were frequently found along roads, often found around natal grass as well. And also I was hopefully confident I could germinate them based on previous studies. Didn't always work out, but yeah. Yes.
You kind of touched on it a little bit, but what are some of the things you'd like to see come from this study? Like further research, maybe applications and all that, but what questions would you like asked and looked into?
Yeah, so I think a little bit more application could be interesting just because my field results were kind of like not wonky, but like, they weren't what I expected. So I would love to keep like piecing that apart, seeing if I can figure out like what's going on. Yeah, I know like road modifications is like one of those things that's like sometimes they're necessary. Like, I mean, driving on Archbold, like sometimes that sand is very deep and there is just like a hole and like you do need it to be fixed. But trying to find a balance, I think would be nice, like something like that coming out of this project would be cool. Yeah. Yes.
Do you think that there would be any difference in the success of the native plants in a higher pH environment, like a modified area without the presence of the tall grass? Like, do you think the pH is really the limiting factor here versus the tall grass competition?
For the native species, probably not so much. I mean, I didn't find a significant effect on soil pH for my germination or my biomass of the native species. And we do find it along the native species along the road. So I would guess that the pH is less of an effect on limiting the native species than simply just the presence of the natal grass.
So because the natal grass is like a generalist that can survive better in that
Yeah.
higher pH, and that's blending out all the native species basically.
Yeah, that would be my guess. Is yeah, the natal grass, it germinated a lot earlier. I didn't show that specifically, but I did find like I'm just looking at my experiment, like the tall grass germinated a lot quicker, like they grew a lot faster. And so if something like my study where like all of the seeds just drop to the ground and are like all of a sudden about to germinate, like the tall grass would get like the advantage of like early and then like growth. And so I think it's definitely a factor of just like tall grass goes and it just blankets. Yeah.
Any questions? Also just say that this is a very timely one because as Emily was starting this, we've been having a lot of spirited discussions among directors about whether we should be adding any road modifications to our Archbold roads because some of them are in very poor shape, as you know. Anyway, we have some refreshments out there and let's get Emily a round of applause.